So hello everyone in the meet. I guess we'll wait for 5 minutes for someone else to join and then we will start the session. Till then, if Niranjana, if you have any specific questions to ask, maybe we can address those. If not, then we will wait for everyone else.
Sir, I kind of didn't understand DNA microarrays, so can you elaborate on that?
Yeah, we can go ahead and discuss that. So DNA microarrays. So to start off, say you want to identify the number of genes expressed in two different separate conditions. Say you have a healthy condition and you have a disease condition. So how will you go about doing that? The first and foremost thing that you have to understand is that for gene expression to occur, mRNA should be made. That is, transcription is the first step. And essentially, any kind of analysis of the gene expressed during either of the condition has to start with mRNA. So, but mRNAs are RNAs as such as very unstable. So most of the studies are done with cDNAs. That is, we use what is known as a reverse transcription enzyme, reverse transcription process using the reverse transcriptase enzyme, and convert the mRNAs that we have in our samples completely to cDNAs, which are complementary single-stranded DNAs. Now that is what is the starting point of the entire experiments with microarrays. You have a sample which is healthy condition, say, and you have all the total RNA extracted from that, and you make cDNA from it. And you have a disease sample from which you again extract RNA and make cDNA out of it. Now the entire logic of it is that you have to understand, you want to know which cDNAs are essentially differentially expressed, that is expressed in one condition and not the other. You are not really interested in the ones that are expressed in both, because those are basal level expressions. So one of the techniques that is used to understand the differential expression between two different states is DNA microarray. What you do is that you have essentially a chip on which you have immobilized uh, complementary DNAs to every one of the cDNA possible in your sample. So every one of the cDNA that could have been, doesn't matter if it is the disease state or the healthy state, you do just make the complementary DNAs to everything, and you immobilize them on different wells in the entire chip, which is essentially like via microfluidics you imprint. That is done. Now what you do is that while making your cDNA in the healthy state, you incorporate a fluorescent labeled nucleotide. Say you are you essentially tag the healthy state of the healthy state cDNAs with one fluorescent label, say Psi3, and the unhealthy state or the disease state with another different fluorescent tag, such as Psi5, the which essentially is green and red. So your healthy cDNAs will look like green, and your disease state cDNAs will look like red. Then what you do is that you take your healthy sample and you put it onto the cDNA chip, the microarray, and you let them immobilize, and then wash off whatever is extra. And then you visualize. What you will see is that in the entire, say, the huge number of wells that you have, you will see green spots light up. Those are the positions in which the hybridization has occurred. So there you can say that in my healthy state, the particular well that has that is lighting up green is the cDNA that was being expressed, or the mRNA or the gene that was being expressed in my healthy state. You do the same with your disease state. You take all the cDNA in the disease state, which is marked red, put it onto the microarray, let it hybridize, and then wash off whatever is extra. And then you try and look into what exactly, whichever regions are lighting up red. The ones that are lighting up red will have complementary cDNAs to it, and you can say these were being expressed in my disease state. Now what you will do is that you overlap both the images, and when you overlap both the images, you will have some spots that will only occur as green, some spots will only occur as red, and some spots will occur both as green and red, and after overlapping they will occur yellow. So essentially what will happen when you align it perfectly, essentially the cDNA, the wells are complementary in both the microarrays. So what essentially is occurring is that if the genes are being expressed in both healthy and disease condition, they will come up as yellow. If only in healthy condition, that will be green. In only disease condition, then it will come up as red. Now you analyze and you see that essentially you're very interested in the genes that have been turned off. That is the green ones, only green ones that are only expressed in healthy condition, and the genes that have been turned on, that is red, which is only expressed in the diseased conditions. You're not very interested in the yellow ones. And since you have made the microarray, you know which well is complementary to which gene. So say you in one disease condition, you in the healthy condition you had a gene X which is still being expressed as green, and you see another gene Y which is now being expressed in red. So you can say that now you can pick and choose and study those particular genes and understand what their effect is on the diseased or the healthy state. Was that clear, Niranjana?
Yes, sir. Thank you, sir.
Okay. Anyone else, if they want to ask anything regarding the week 3 session, then you can go ahead and ask. Otherwise, you can probably start in another 2-3 minutes. So let me ask something instead. Like ask something back. So there has been a new technique that is now essentially the standard in the field. If I want to understand the gene expression or the RNA expression in a particular sample. And, can anyone tell me what is the new technique that is being used, like that is now the standard in the field? Like DNA microarrays were extensively used, but they are a bit older now. Like DNA microarrays are not very used nowadays. Can anyone tell me what new technique has overtaken it as the key to understand the RNA expression or the gene expression in a particular sample? You can guess, at least try guessing if you know. Like the question is, let me put it into the chat box. Here is the chat box. Maybe by the end of it, someone can tell. Like end of the entire talk, someone can at least go and Google and try and tell. So since we have a lot of people, we will probably start the session. Yes. I hope the PowerPoint is visible. Yes.
Yes, sir.
Okay. So let us get started today. Hello everyone. I welcome you all to the week 3 bioengineering PMRF-NPTEL session on the course Bioengineering and Interface with Biology and Medicine. The PMRF-NPTEL sessions are sessions in which we hold interactive live sessions with the students of the course and a PMRF fellow such as myself, where we go through the previous week's content, of the course and understand how exactly to tackle various assignment problems posed on them. We essentially solve the previous week's question assignment questions along with the students in order to make them better understand the course material and how exactly to tackle the various assignment questions given to them. Let me introduce myself. My name is Anubhav Chatterjee. I'm a PMRF fellow and a PhD scholar at the Developmental Neurobiology Lab, Department of Biological Sciences and Bioengineering, Indian Institute of Technology, Kanpur. This week, we are going through the week 3 lecture course lectures, in which we have essentially understood about advanced DNA technologies in order to study gene expression, and we will be solving various questions regarding those. So we have some answers to a previous question in the chat. The question was, what new technique has become standard to study gene expression or transcriptomics of a sample? The answers that were given are microarray and RT-PCR. So microarray is something that we just discussed at the start of the session. So microarrays, as we just discussed, are very nice techniques to use, but that has now been overtaken by another new technique. So it is not DNA microarray that the answer we are looking for, and Misa says it is RT-PCR. So RT-PCR has a separate use than the that understanding the transcriptomic profile or the RNAs or the genes being expressed in a sample. So let me ask this. Anyone can just unmute and tell me, in for RT-PCR, what extra information do you need than doing a normal or than doing a microarray? Like what information, for if I wanted to say I want to do RT-PCR of my sample, what explicit information should you have? Like you can just go ahead and tell me what exactly information you think you should have before setting up a PCR reaction. Yeah, primer. Chan is correct. We need to know the primer. So in order to know the primer, that just tells you that you have to know the gene sequence, because you cannot make a primer specific to a gene without knowing the gene sequence. So you cannot essentially do a gene profile analysis or gene expression analysis without knowing every one of a sequence of every one of the genes present in the cell. So if you already so you started out to understand which genes are being expressed. So you were going into the unknown with what genes are being expressed. And RT-PCR is a technique that expects you to already know the gene sequence of the genes you want to amplify. So RT-PCR is not the one that we use to study the gene expression. RT-PCR is instead used to understand the quantitative amount of genes being expressed. So you already know the gene that is being expressed and you want to know in how much quantities it is being expressed. So the technique that I was talking about, the new technology that is now the standard, is RNA sequencing or RNA-seq. Let me put it into the chat. RNA-seq. This is what nowadays is being used. So maybe so this does not require you to understand to know the gene sequence. It just uses a oligo DT primer in order to amplify your entire mRNAs and sequence them. So this is like a very powerful technique in order to understand the gene expression. So maybe you can I would encourage everyone in the meet as well as who will look into the video on YouTube to go into literature or just Google the term RNA-seq and learn how exactly what is going on. Like this is now the standard technique that is used entirely everywhere in the field if you want to study gene expression. So we also have someone new that joined. So we will start with the question answer session. So this is the first question we have here this evening. And this evening we have Prakhar, who is a MSc student who is working on on the human gene insulin and wishes to produce the protein for therapeutic purposes. So he has the gene. So that means that he has the sequence, and he wants to make a functional protein out of the gene. We have to help him purify the protein, where he has used the N-terminal his tagged gene to facilitate the protein purification process. So N-terminal his tag is something that was discussed in the course in the lectures, but we'll go through it in the following questions. So guide Prakhar to choose the correct expression host. Which of the following features should be considered to choose the correct host? So which of the various features do you think is appropriate when Prakhar is going in search of which host can be chosen? So the options are length of the gene, presence of introns, post-translational modifications, and toxicity to the host cells. So this is only one option that is correct. So you can go ahead and put the option that you think is correct in the chat box. So we already have two answers. Meeta and Niranjana both say it is post-translational modifications. Okay, so anyone else that wants to try and answer this? You can just put in ABCD in the chat box. Anyone else that wants to try and answer for this question? Smruti also says it is C, post-translational modifications. Okay, so Archana also says it is C, post-translational modification. Excellent. So let us go ahead and understand what exactly we are looking for here, and we will get back to the answer of the question. So when we are talking about gene expression is not just transcription or translation. Gene expression has a lot of steps, and some of the steps is like the steps are given here. The first step of gene expression is DNA to RNA, or the DNA to nuclear RNA, which is called transcription. And gene expression can be regulated at the transcription step, which is known as transcriptional regulation. This is followed by RNA processing to make a mature mRNA or messenger RNA via the process via the process of via the process of splicing and various other steps, which include the 5' capping or poly A addition at the end of the mRNA. So this is RNA processing, and any regulation that is done between the original transcript to spliced mature mRNA is known as post-transcriptional regulation. Next, the mRNA that was made is transported from the nucleus to the cytoplasm where it will be translated. And here also the nuclear pore complexes can regulate and stop the trafficking of mRNAs, and here also regulation can be done. Next, after we have the mRNA in the cytoplasm, we have a ribosome that attaches to the mRNA, and then we can have either translation of the mRNA to protein, or mRNA can be regulated at this step also. So it can be degraded or silenced via various mechanisms, and this is also part of post-transcriptional modification, post-transcriptional regulations. Next, we have translation. The ribosome sits on top of the mRNA and forms a protein. Here also regulation can occur, which is known as translational regulation. And finally, from the after a peptide is synthesized, it undergoes post-translational modification to form an activated protein. And post-translational modification is also a very important step which regulates the activity of the protein. And if the protein is not correctly folded, so one of the post-translational modification is folding, so it should acquire the appropriate quaternary structure, which is the active form, and have various other moieties added to it, such as glycosylation or phosphorylation or various other steps. If any of this does not occur properly, then we have a deactivated protein or non-functional protein which is promptly degraded, and that is known as post-translational regulation. So the entire flow chart from DNA to activated protein is gene expression. It is not a one-step or two-step process. It is multi-step process. But during cloning or gene expression in other prokaryotic systems per se, the important step that we want to look into is post-translational modification. The major reason is that many of the post-translational modifications that occur in eukaryotes are not replicated in prokaryotes. So even if we have taken in every other precautions, our protein might not get folded or have the proper post-translational modification to be active, if we do not choose our host very properly. So this was the entire slide on gene expression. Is this one clear? Because next we have a video on various post-translational modifications. Does anyone have any question regarding this? I guess not. Okay. So we move on to the video. So in this video, we'll essentially look into how post-translational modifications work and why they are important.
In the last few decades, researchers have discovered that
So you can hear the audio, right?
the human proteome is vastly more complex than the human genome.
Okay, we have a question in the chat from the previous slide, so we'll probably address that before. So what is the original RNA transcript? So when our process of transcription occurs, the first RNA that is produced is known as nuclear RNA. And nuclear RNA, if you will recall, has introns and does not have other characteristics of mature mRNA such as the 5' cap or poly A tail. It is just the copy of DNA converted to RNA form. It does not have any other changes or processing done to it. That is known as the original RNA transcript. In order for it to the next step in RNA maturation is splicing followed by 5' capping and poly A tail addition. So then what you get is known as a mRNA. Before that, it is known as the human nuclear RNA, or here in this diagram, it is given as original RNA transcript. Did you understand, Archana? Capping. Okay. Okay. So in eukaryotes, in during eukaryotic gene expression, when our RNA has been spliced, we what we have is that at the 5' end there is a 7-methylated guanosine residue, a base attached in the opposite direction, essentially, which is known as the 5' cap. That is attached for the only reason so that no exonucleus, 5' exonucleus can like recognize the 5' cap and start degrading the mRNA. So mRNA, as you know, is very, very like labile. It can be easily degraded by anything in there, and most important enzymes that degrade RNAs are RNAs, like RNAs, RNA, AAC, like DNAs, RNAs. So these enzymes are present everywhere, in the cell, in the air, everywhere, and that is very dangerous to RNA work that most of our labs do. And what they do is that they go ahead and chew up the RNA so that you do not get any translation or any other step. There is no question of that. So what mRNA needs to be protected so that it is not easily degraded till it has been translated. So 5' capping is one of those steps. We attached we attach a 5' guanosine cap to the mRNA that protects the mRNA from 5' exonucleases that recognize the last 5' nucleotide and start cutting up the DNA. So 5' cap prevents that. Similarly, the poly A addition in eukaryotes is also due to that only. So that a 3' nuclease, even if it starts cutting the mRNA from the other end, it starts cutting the A tail first, which does not have any important information. So these are protective mechanisms of mRNA. So if you go ahead and yeah, okay, Archana says she has understood. Okay, so if you want to know more about this, you maybe can look into eukaryotic gene expression. So there are a number of books that have these. So Leninger is one of the major important biochemistry books, which have a lot of information on these. But you can also watch any YouTube videos and try and understand. So if that is done, we will move forward. And yeah, please tell me if you can hear this audio of this particular video. Which is very fun.
In the last few decades, researchers have discovered that the human proteome is vastly more complex than the human genome.
Yes, sir, we can hear it. Okay, nice. So let me start and we can observe the video.
In the last few decades, researchers have discovered that the human proteome is vastly more complex than the human genome. Post-translational modification of proteins increases this functional diversity of the proteome, which is the entire set of proteins expressed by a genome at a certain time. The fact that there are way more proteins than there are genes demonstrates that single genes can encode for multiple different proteins. And the increased complexity of the proteome as compared to the genome is facilitated by many different mechanisms, including post-translational modifications, which help to regulate localization, activity, and interactions with other cellular molecules. These modifications can occur at any time during the life cycle of a protein. Many proteins are modified right after translation is completed, and these modifications help to mediate folding into proper conformation, increase the stability of the nascent protein, or help to localize this protein to distinct cellular compartments, like a shipping label. Other modifications occur after folding is completed and a protein has been localized to its proper cellular locale. These modifications serve to alter the biological activity of the protein, either by activating or inactivating catalytic activity, for example. Some other modifications serve as markers that target a protein for degradation. And besides chemical modifications, proteins can be modified via cleavage or proteolysis. The key thing to remember is that the proteome is dynamic and responsive to all sorts of changes in stimuli, and post-translational modifications are a common mechanism for regulating these cellular activities. Now, there are many types of post-translational modifications, so I'll go over only the most common ones here. And let's start with methylation. Methylation involves the transfer of one carbon methyl groups to amino acid side chains by methyltransferases using S-adenosylmethionine, or SAM, as the primary methyl group donor. This can neutralize a negative amino acid charge when bound to carboxylic acids and leads to increased hydrophobicity of the protein. A well-known use for methylation is epigenetic regulation of transcription. Histone methylation and demethylation can alter the availability of DNA for transcription. Another type of protein modification is acetylation, specifically to nitrogen atoms on a protein, so N-acetylation, which occurs as the nascent protein is being translated. The N-terminal methionine on the growing polypeptide chain is cleaved by methionine aminopeptidase and then replaced by an acetyl group donated by acetyl-CoA via an enzyme called N-acetyltransferase. Up to 90% of eukaryotic proteins are acetylated this way, though the biochemical significance of this modification remains to be known. Acetylation also occurs on the lysine residues of histones via the action of histone acetyltransferases, and this is also used to alter transcription in a similar manner to that of methylation. The acetylation of histones helps to promote transcription by reducing the chromosomal condensation around these proteins. The next post-translational modification that can occur to proteins is called glycosylation, and it is one of the most significant types of post-translational modifications because it has implications on protein folding and conformation, distribution, stability, and also activity. Glycosylation involves the addition of a diverse array of sugar moieties and ranges from simple monosaccharide modifications of transcription factors to highly complex branched polysaccharide modifications of cell surface receptors. These carbohydrates can be added to the nitrogen atom in the side chain of asparagine residues, those are N-linked, or to the oxygen atom in the side chains of serine or threonine residues. These are O-linked. These types of glycosylation changes form major structural components of cell surface and secreted proteins. The next modification, lipidation, is a post-translational modification employed to target proteins to particular membrane-bound organelles, such as the endoplasmic reticulum, Golgi apparatus, or mitochondria. It is also used to target proteins to endosomes, lysosomes, and the plasma membrane. Two types of lipidation modifications are GPI anchors and S-palmitoylation. C-terminal glycosyl phosphatidylinositol, or GPI anchors, help to tether proteins bound to the plasma membrane of the cell surface. These hydrophobic moieties are prepared in the endoplasmic reticulum, where they are added to nascent proteins and used to localize cell surface proteins to cholesterol or sphingolipid-rich areas in the plasma membrane. S-palmitoylation involves the addition of 16 carbon-long palmitoyl groups to thiolate side chains of cysteine residues. This modification adds a long hydrophobic chain that can be used in a similar manner as a GPI anchor. It helps to anchor proteins in the hydrophobic cell membrane. Next is ubiquitination, which is a protein modification used to target proteins for degradation. Ubiquitin is a polypeptide consisting of 76 amino acids that is attached to lysine residues of target proteins via the C-terminal glycine of ubiquitin. Polyubiquitinated proteins are recognized by the 26S proteasome, which is an enzyme that catalyzes the degradation of the protein and the recycling of the ubiquitin. Next up is one of the most common post-translational modifications that you'll come across, and that's phosphorylation, which is a reversible modification that occurs principally on serine, threonine, or tyrosine residues and is used to regulate proteins that play a role in a vast array of cellular processes, including signal transduction pathways, the cell cycle, cell growth, and apoptosis. Protein kinases are the enzymes that help facilitate the phosphate group transfer, and phosphorylases help to remove them. And lastly, enzymes called proteases may remove amino acids from the amino end of the protein or cut the peptide chain in the middle, a process known as proteolysis. One example of this is the peptide hormone insulin, which is cut twice after disulfide bonds are formed, and a propeptide is removed from the middle of the chain. The resulting protein consists of two polypeptide chains connected by disulfide bonds. Proteases also play roles in cell signaling, antigen processing, and apoptosis. In the last few decades,
Okay, so that is enough for the video. Yeah. So essentially, this is a nutshell video for how exactly what different types of post-translational modifications are and what are the advantages. And these are what we look for in the while choosing the host organism. So let me go through them very quickly so we understand what exactly is going on. So post-translational modification is essentially changes done to the protein after it has folded properly. That is, it has achieved its quaternary structures, and now you pick and choose amino acids to somehow modify them. The first one that has been discussed is methylation. Essentially, you add a methyl group to a accepting amino acid, and you essentially the amino acid will now have a CH3 attached to its functional group. One of the important uses for it is methylation of histones in DNA, and if region of the chromosome is highly methylated, that there is high histone methylation in that region, then that region is suppressed. There is no gene expression there. The opposite is acetylation, where you add an acetyl group to a particular amino acid in the histone chain, and adding a acetyl group to the histones in the chromosome will allow an increase in transcription in the region where this acetylation is going on. The third post-translational modification is glycosylation, in which essentially you add sugar moieties. This can be glucose, like obviously we know sugar moieties are carbohydrates, which can be normal glucose like as simple as glucose to as complex as sphingolipids or phospholipids, which not sphingolipids or phospholipids, that is lipidation, various branched carbohydrates, which essentially change how exactly the folding can occur. The fourth is addition of various lipids such as sphingolipids or phospholipids, which can target the particular protein to the membrane and act as cell membrane receptors, or change the way how exactly the hydrophobicity of the protein is. Next is ubiquitination. If a particular protein, the post-translational modification is ubiquitination, so essentially the protein is now being marked for degradation. So the protein will now be degraded if it is ubiquinated. So, and that degradation is done by the Proteo26S proteasome. Sixth is phosphorylation. So activation of proteins, especially enzymatic activation of proteins, is done via phosphorylation. So essentially, a phosphate group is added to a protein, and that signals that the essentially the protein is now active to do its whatever its biological function is. And finally, we have proteolysis, in which another peptide, like another protein, comes along, which cleaves amino acid from the protein and somehow changes its configuration. Major important example is insulin, which, as in the video showed, that insulin is in the beginning a single chained peptide, which is then cut on two ends to form two different separate peptide chains, which are linked with double disulfide bonds, and this is known as proteolysis. This also has implication of uh, the protein function and how exactly it will act in its biological aspect. So these are the various post-translational modifications. Does anyone have any questions regarding this? So probably not. So essentially, well, what is the takeaway from the slide is there are various post-translational modifications that are very important for various functions of the proteins. And if we want to clone a protein and express a protein in a separate host where it is not usually expressed, then we have to make sure somehow to incorporate these various post-translational modification into the protein. That is essentially what the takeaway from the slide is. So that was that. And since we are talking, our friend Prakash, right? Yeah, he is studying insulin. So we'll just go through how exactly insulin production works. So, as I said, insulin is a single peptide to start with. It folds and forms two disulfide bonds, and one part of the peptide is broken off to form two separate chains connected by two disulfide bonds. So, this is the active form of the insulin, and it all starts from another form which is known as pre-pro insulin. So, that is inactive. But how exactly does like recombinant insulin get produced? So there are a lot of modifications that are taking place here. So how exactly do we make sure that this occurs normally in the prokaryotic systems that we express the protein in? So it is done very smartly. What is done is one part of the peptide, the upper chain, is produced, which is known as the A chain, produced in one E. coli or one bacteria, and the B chain is produced in another bacteria. And essentially both are combined and allowed to form the disulfide bonds only. So no question of cleavage or specific cleavage at particular sites or anything. Just form the A chain, form the B chain, join them together with disulfide bonds. And that is how active insulin or active recombinant insulin is produced, which we see mostly sold on the market. So, according to the question, guide Prakar to choose the correct expression host. What should be considered the most? The most thing that is should be considered is post-translational modification in the particular host. If the various post-translational modification that we need are being done in the host or not. So, is that understood? Does anyone have any questions regarding this? It's fine, right? Okay. So we move forward to the second question about this entire journey with Prakhar. So Prakhar has selected the E. coli system for protein expression, but when he grew the culture with the PET28A expression vector, he could see low growth of the bacteria. So he had chosen vector 1, in which he has seen low growth. He transformed the recombinant vector in the expression host with PLYSS, so PLYS plasmid, and the transformed plates are shown. So these are culture plates with the various columns. So the white are the columns. Which of the following observation is false? The options are: A is equalized chain with gene of interest and P-list plasmid. B is equalized chain with gene of interest and P-list plasmid. P-list helps in debilitating the leaky expression of gene of interest, and protein product of gene of interest is toxic to the host cell. So this is only one option type of correct answer. If anyone has any guess as to which one is correct, then you can go ahead and answer in the chat box. So this question does not essentially have to do anything with the P-list plasmid or any information given here. Essentially what is happening is our friend Prakhar first did it in PET28A vector, and he didn't see much plasmid proliferation, like many plasmid colonies, bacterial colonies, and he then went to P-list plasmid, which should have higher number of colonies, and thus this is the B plate is what he got. So the A plate is where he had the PET28A vector, and the B plate is where he had P-list vector. So the first option is the false one. That is, A is the equalized strain with gene of interest and P-list plasmid, because P-list should have a higher number of colonies. That is, it is the B that is the true answer. So B is where the P-list plasmid and gene of interest is, and A is the one where the PET28 vector is, and that is A is false. We move on to the next question. So third question is, Prakhar successfully selected the PCR confirmed colony of transformed P-list bacterial strain for protein expression and purification. He lysed the cell and extracted all proteins. The desired protein is histone. So which of the following chromatographic techniques will be useful for protein purification? The options are reverse phase chromatography, ion exchange chromatography, affinity chromatography, and size exclusion chromatography. Only one option is correct. This is something that we have that has been discussed in the lectures as well. So only one option is correct. You can go ahead and put it into chat box, whichever you think is correct. Niranjana says it is affinity chromatography. Everyone else, please try and answer this. So Archana says it is B, that is ion exchange chromatography. So yeah, so anyone else that wants to try and answer this? Shruti says it is A, reverse phase chromatography. Okay. Uh, so let us try and understand what exactly should be the. Amardeep says it is C, affinity chromatography. Okay. So let us try and understand what exactly is going on, and let us come back to the question and try to answer. So first, the major thing is what exactly is chromatography that we are talking about. So chromatography is an important biophysical technique that enables the separation, identification, and purification of the components of a mixture for qualitative and quantitative analysis. Essentially, if you have a mixture of separate things, various things, and you want to separate them based on various characteristics, could be the boiling point, could be the weight, could be the charge, anything, that can be done using the process known as chromatography. So chromatography is based on the principle where molecules in a mixture applied onto a surface or into a solid, and a fluid stationary phase is separating from each other while moving with the aid of a mobile phase. The factors effective on this separation include molecular characteristics related to absorption, which is a liquid solid interaction, partition, or affinity or differences among molecular weights. Essentially, what chromatography is, I think example that will be more to the heart, like something that is very well understood, is that if you have ever seen uh, a sand filter, if your a sand filter is essentially a tub filled of sand, and you what you take is you take muddy water, and if you put it into the sand filter and allow it to go to the bottom, what will happen is that all the sand particles will get stopped in the sand layers, and only pure water will come out after all the purification. Have you seen such things? Yes, no, maybe. You can put it into the chat if you understand what I'm trying to say. Yeah, Niranjana says yes. Anyway, everyone else also understand what I'm trying to say. So sand filter is essentially a layers of sand. You put water muddy water on the top, and what you get in the bottom is pure water. Yes, everyone understood what I'm trying to say. Yeah, Amardeep also said this. I guess. Yeah. Okay. So that is clear. So that is essentially what chromatography is. Chromatography is when we want to separate various if we when we want to separate various things based on their size or shape or charge, we allow them to pass through a solid phase, which in our case is the sand in the sand filter, which will stop some things and allow others to pass, and a liquid phase or a mobile phase, which is essentially the muddy water, which forces the entire thing to go down. These are the two phases. So originally what we have is the mobile phase contains the mixture, what the things that we want to separate, and we put it into the solid phase, and the solid phase is the one that is doing the filtering for us and allows only some to pass and others to pass very very slowly. That is essentially the entire principle of chromatography. So here in our example in the schematic, what we have is that instead of the sand we have a silica column. So silica gel is just a neutral stationary phase substance that has holes in it, and we add the solvent or mobile phase that contains our mixture of things. So in this particular example, we have three separate colored substances. One is colored blue, one is colored green, one is colored orange, and everything is mixed. Not like the diagram here showing three bands in the starting, but a mixture at once. What happens is that once I continue to pour the mixture on the top, one of those layers will be will have a higher molecular weight or higher size. And what happens is due to its higher size, it starts moving faster and goes essentially not due to higher size. What happens is something that we will discuss later, but essentially due to some characteristic of the silica column. And I have lost my. Am I audible? Hello? Hello? Am I audible?
Yes, sir.
Yeah, thanks. So, what I wanted to say is that you have a mixture of separate colored components. You add it onto the silica column. Due to specific characteristics of the silica column, it starts separating those. So, what happens is that after you have filled the silica column, you just leave it, and the the colored components will start like flowing out one after the other. So essentially, if you have one tube, you can essentially capture the orange colored substrate once, remove, go to the second tube when the green colored substance starts forming, if flowing down the column, catch it and then remove, and then lastly you do it the same with the blue colored substrate. Essentially, what you're doing is that you're separating based on specific characteristics of the mobile phase and the solid phase, and using it to separate out a mixture of various components. So you had mixed analyte, and now you have different bands that are collected in the separate flasks, and that is why it is known as chromatography. That is separation based on color. But obviously, in our case, like advanced forms that are used nowadays do not have anything to do with the color, but the name has remained. Essentially, you pass your substrate through a column, you separate everything inside the substrate based on the size or various other characteristics. So this is what chromatography is. Now let us go into the types of chromatography, like how exactly the separation occurs. So we deal with major types of chromatography only here. The first is liquid column chromatography, in which we have a solid stationary phase such as silica or alumina. So liquid chromatography is the diagram we just saw. what it does is liquid phase is mobile phase is liquid and it separates on the basis of polarity. For example, say you had a mixture with negatively charged particles and positively charged particles, and you wanted to only catch hold of the negative charged ones. What you do is, in that case, you use the liquid column chromatography technique, and your silica stationary phase, that is the column, that is positively charged. You pour down your entire, no, that is not liquid column chromatography. Sorry, what that was wrong. So polarity of molecules is essentially similar. It just the because of the size of the particles as well as the charge that they are carrying, some of the particles will be slow while moving through the column and some will flow very fast, and that is how you separate during liquid column chromatography. In case you have the charged thing, that is your mixture contains a negative charged substance and a positive charged substance, and you use a solid resin, yeah, solid column which is positively charged, then it will only specifically go hold on to the negatively charged substrate and the positive will just flow through. And essentially what you have done is you have separated your mixture based on the ionic charge, and that is known as ion exchange chromatography. So if you want to get hold of the negative charge substances inside your mixture, you use a positive column to catch hold of it, which is known as a cationic resin. And if you want to catch hold of the negative charge substances, then you use the anionic resin or negatively charged resin. They will just attach to each other using electrostatic attraction, and you have separated your desired substance out of the entire mobile phase. Another is size exclusion chromatography, which separates based on the size. And the important one that we are looking for here is affinity chromatography. So what affinity chromatography is a bit more advanced than the other ones. What happens is that the solid column or the solid stationary phase has various binding sites to which substances in the mobile phase can bind, and these binding sites specific. And what happens is that only the molecules that bind to a binding site can stay stuck to the column and everything else just flows through. So during affinity chromatography, you have a solid agarose or porous glass beads with immobilized molecules which are your binding site. These can be enzymes or antibodies. And while the liquid phase is passing through, the binding affinity of the analyte molecule that is present in the liquid phase is immobilized to the stationary phase and gets stuck to it. And you essentially separate that particular molecule of interest out of the liquid phase. So this is what affinity chromatography is. And finally, we have gas chromatography, which separates depending on the boiling point difference between various molecules. So these are the major types of chromatographic techniques. Does anyone have any questions regarding this? So does anyone has any questions regarding the types of chromatography? I guess not. So we'll move forward and learn what exactly affinity chromatography is, the one that we need. So in affinity chromatography, especially the histidine tagged affinity chromatography, what we have is that we have a tagged protein that has a 6 his tag. Essentially 6 histidine that are attached to either the N terminal or the C terminal of the protein. Now, a specialty of 6 histidine tag or 6 histidine residues are one after the other is that they can form coordinate bonds with nickel, which is a very interesting phenomenon that we use during affinity chromatography. Essentially, we have our protein with our histidine tag, 6 his tag, and our column has nickel attached to it. So the column is known as a nickel NTA column, which has nickel attached to it. Essentially, the protein, the entire lysates, all the proteins in the cell flow through it, flow through the column, and only the proteins with the histidine tag get attached to the column. and everything else flows through, and you then wash it again and again till every other non-specific protein just washes through, and only your protein of interest, which has binded to the nickel ion, is bound. Then you add something known as an elution buffer, which elutes only your desired protein, like the histidine tag proteins, and you collect it separately from the other lysates, and then essentially you have purified your histidine tag proteins. So the steps are: you take the entire protein out of the cell, put it into the column. In the column, nickel is there that will attach only to the proteins having the histidine tag. You wash again and again till all other proteins are washed through, and you essentially throw that out. Put your particular and then put a vessel underneath your column. Add the elution buffer, and the elution buffer will bring along whatever your histidine tag proteins into your vessel, and essentially you have purified your histidine tagged protein. This is the principle behind affinity chromatography using histidine tagged proteins and nickel NTA column. So, is that clear? Does anyone have any questions regarding this? Affinity chromatography is clear, no clear, maybe. Hmm. Yes, no, you can like just, yeah, okay, fine. So it is clear. So, so we will see how clear it is since everyone seems to understand, because the next question is the follow up on these. So Prakhar successfully selected the PCR confirmed colony of the transformed T list bacterial strain for protein expression and purification. He lysed the cell and extracted all the proteins. The desired protein is his tagged, so he can use the affinity chromatography technique using a nickel NTA column in order to purify his desired protein. So the next question, let's see how well we have understood the affinity chromatography. The opted chromatography was conducted and elute was collected. So essentially you elute you have washed everything and eluted what you think to be your particular protein of interest. Refer to the image help and help Prokha to understand the possible reasons why the elute's profile looks like this. So what we see here is 1, 2, 3, 4, 5, 6, 7, 8, 9 columns. Do not bother with anything else. Just look at the ninth column. And this ninth column is essentially when you elute the protein. What you see is on a protein gel, 4 bands. That is, there are 4 different proteins here instead of the one you wanted. Instead of the one histidine tag protein that you have 4 bands of different proteins. Why is that is the question. Why do you think there might be multiple proteins getting attached to your nickel column and then eluting out, where only you had possibly one NTA, one histidine tagged protein in the entire cell? So the options that are is that N-terminal histag might lead to production of a truncated protein. The elute can be a multimeric protein, bacterial protein with high histidine content, and A and C answers are correct. So this is a one option type of answer. You think and think back to our discussion on nickel NTA columns on affinity chromatography. We are back on that slide. Think why when we elute, may we get more than one protein? So the options are that our histidine tag is causing the protein to be truncated, that you are getting a smaller protein. Second was that second one. Okay. Second was that the elute can be a multimeric protein, that there are multiple subunits to the protein. Third is that there are bacterial proteins with high histidine content. And fourth is that A and C are correct. You think why there can be multiple protein bands in the gel when we elute, and then try and answer this. Let us see how well have we understood affinity chromatography. Amardeep says C. That is, the bacteria has high histidine content proteins. Okay. So, chalo, let us see everyone else try and answer this. So here are the options. So N-terminal histag might lead to a malproduction of truncated protein. The elute can be multimeric, bacterial protein with high histidine content, or A and C both are correct. No one else wants to try at least. Niranjana says D. That is A and C. Okay. Anyone else? Okay. So Amardeep, can you unmute yourself and let us know why do you think it should be C? Like, what was your thought process? Hello, Amardeep. Like, you can even put it into the chat if you think that will be easier. Why do you think is it a C, bacterial protein with high histidine content? Okay, maybe Niranjana, do you think you can let us know why do you think it should be A and C both?
So I thought maybe because lanes 5, 6, 7 show very less expression, maybe the proteins are truncated, and as a result of which they are no longer expressed. I mean, they cannot be seen in the chromatography profile. So the first option. The second option, because they are indeed expressed in 2, 3, and 4, so bacterial protein might have high histidine content.
Okay. So this second, like the C option thinking was correct. That bacterial protein with high histidine content. So essentially, what is happening is in the entire process, if we have followed, is that high histidine content. Essentially, the 6 his tag is histidine containing protein that binds to nickel. So any other bacterial protein, if it is high, has high histidine content, then it can also bind to the nickel, and that can be one of the answers due to which bacterial protein is like another protein is being detected. So bacterial protein with high histidine content, that is correct. N-terminal histag might production lead to the production of truncated protein. So we have to understand why, the other reason due to which there can be multiple bands. Say if your histidine tag was causing some kind of folding defect in the protein, then, as we just saw in post-translational modification, it will be lysed or cleaved into smaller parts that the cell doesn't want a non-functional unfolded protein going around. So what it does is it goes ahead and cleaves the entire protein into parts. When it cleaves the protein into separate parts, you have a smaller peptide which still has the his tag attached to it. So what that does is that it creates another band on the chain. So you have a smaller peptide which still has a his tag, And when you run the entire thing, you will get the original protein which had the his tag, and the smaller truncated protein that was cleaved off and just has some part of the protein plus the histidine tag. And that is why you can have, yeah, we have another H tag can bind to many proteins containing histidine. Yeah. So that is the second one. So the first one was because histidine tags is there, that might result in cleaved proteins which still have smaller fragment of the protein and the histidine tag, and that will run below your original protein. And obviously, as Amardeep has pointed out, the histidine tag sequence can bind, like his tag, or the essentially the nickel can bind to other proteins that have high histidine content. Like you have enough histidine, and it will nickel obviously does not discriminate and binds to the histidine via coordinate geometry, a coordinate chemistry. And when we elute out, we will have the other bacterial proteins as well that have high histidine. So C is correct and A is also correct. So the actual answer is D, that is A and C. Can you explain option B too? Yeah. So the elute can be multimeric. So what is a multimeric protein? Multimeric proteins means that they have separate peptide chains that come together to form a quaternary structure. A very nice example is hemoglobin. Hemoglobin has the alpha chain and the beta chain, and two alpha chains and two beta chains come together around the iron to form the quaternary structure. So four separate units are there. So that is known as a multimeric protein, which has separate peptides that come together to form the entire quaternary structure. Now, what happens is that when you have put in your histag, that is at the end of a gene, or that is at the end of a gene. So that gene will form one mRNA with the histag, that will form one peptide with the histag, and that one peptide can only form one subunit of your, you will only form one subunit of your multimeric protein. So even if four separate individual subunits are coming together to form a entire multimeric protein, when you run on the gel, only your his tagged subunit will be detected. You will not detect a bigger four complex protein, which will like, it will not be like that if the peptide is part of a bigger protein, that the entire bigger protein will also be detected on the SDS page. It will not. Only your smaller subunit will be detected. So even though your protein has separate subunits that are being attached to form a functional quaternary structure, when you run it on the gel, only the part that has the his tag will be detected, and that will only form one band. So formation of multiple bands cannot be explained with the elute being a multimeric protein. If that is okay, or I have to draw and make you understand. Oh, you understood. Okay. So I can even draw it on the whiteboard and try and explain if you want, or otherwise it is okay. Yes, no, maybe. Okay. Yes, understood it. Okay. So that was this question. So the answers are A and C. So is this okay with everyone? Because this is a very, like, affinity chromatography is a question that comes up again and again and again in GATE and every other CSIR exams, and that is very simple to answer if you know the procedure of how exactly to tackle it. Is it okay with everyone? Oh, yes, Boldo. Okay. So the next question. Prakhar wishes to confirm each band if it contains the desired protein of interest. Help him to select the correct techniques to confirm the correct band. So what has happened is that Prakhar went and saw that he now has four bands. So how exactly does he know that what is his correct protein? It can be a bacterial protein that came out. So what techniques can he use in order to identify that it is his protein of interest, that the protein he wanted to purify? The options are mass spectrometry, Western blotting, microarray, and ELISA. So this is more than one option correct because it's a box type of answer. So more than one option is correct. You can click 1, 2, 3, or 4, depending whichever you think is the correct one. So depending what you think is the answer, you can go ahead and put it into the chat box. Which technique can be used by Prakhar in order to identify that the purified protein is his protein of interest? Amardeep says B, Western blotting. So more than one option is correct. So B could be a correct answer, yes, but you can even choose more than. So there can be more than one option that is correct. B and D, Western blotting and ELISA. Niranjana says it is mass spectrometry and blotting, that is A and B, and Amardeep says it is A, B, and D. Mass spec, Western blotting, and ELISA. Okay. So anyone else that wants to try and answer this? Okay. So we will go into the solution slide and try and understand which technique it can be. So the first technique that we are discussing here is mass spectrometry. So this is the discussion we had in our very first week session. So we will go through it relatively faster this time. So mass spec is a analytical tool that is used to measure the mass to charge ratio of one or more molecules present in the sample. And mass spectrometry is commonly used as a high throughput tool for studying proteins. What is done is you take your protein, you can break it down. So essentially what mass spec does is that mass spec consists of a tube that has an electric field across it. You throw your peptide or molecule across the tube, and depending how much time it takes to get to the other end, you can calculate what is known as a mass to charge ratio. That is the mass of the particular molecule divided by the charge it has. This can give you an idea what exactly the molecule was, because mass to charge ratios are more or less conserved, and you can essentially figure out what exactly the molecule was that you threw. So if you threw a lysine, depending on what peak you get on a mass to charge ratio graph, you can understand that yes, this was a lysine. Similarly, you do it with 2, 3, 4 amino acids. You throw asparagine in there, you throw a valine in there, and depending on to the mass by charge ratio, you figure out what exactly the amino acid was. This is what this funda is used in mass spectrometry to understand the sequence of the particular protein that was thrown, like put into mass spec. What exactly you do is that you first digest the protein, form peptides, put the peptides, you separate out the peptides using liquid chromatography, just something that we just discussed. After separation of peptides, you run the first mass spec. The first mass spectrometry separates out the peptides depending on their mass to charge ratio. You essentially have, after the first mass spectrometry analysis, you have peptide 1, peptide 2, peptide 3, peptide 4, peptide 5, separate peptides. After what you do is that you break down the peptides to amino acid level, and then you run that through mass spec again, and this time around you identify the amino acid sequence. Then you put together the amino acid sequence to form the peptides, and to put together the peptides to form the proteins. And this is done using a database search. Essentially, you calculate everything, and this gives you, after peptide scoring, what exactly your original peptide was, and you then align the peptide with your database and figure out that you are getting one sequence that is common to a part of N terminal part of protein 1, and second sequence that is common to the C terminal part of protein 1, and then you can say it was protein 1 that you actually threw into the mass spec analysis. So essentially, in this particular study, glyceraldehyde 3-phosphate hydrogenase was found out using this technique. This is entirely known as the LCMS or liquid chromatography tandem mass spectrometry workflow. Lot of words. Essentially, you use the mass by charge ratio in order to identify which protein you had to begin with. That is known as mass spectrometry. Anyone has any questions regarding this? If yes, we can delve a bit deeper into this, or if not, we can just go on. Yes, no, maybe. बताइए. Everyone understood what mass spec is. Essentially, it is a technique where you just send your mixture of protein sample. They will run it through a liquid chromatography, followed by first mass spectrometry, second mass spectrometry, and you will have what protein you started. Nothing else. So this can be used as a technique to identify what protein did our friend Prakhar isolate. So if he sends it to mass spectrometry, he gets the sequence, he knows what the protein is, he can say, okay, it was not insulin that I wanted to purify, it is some other bacterial protein. Then I will discard it and go on with the insulin wala sample. And that is what mass spectrometry does. Next is Western blotting. So Western blotting was essentially the gold standard. Western blotting was essentially the is still essentially the gold standard in identifying a specific protein that is present in the sample. So Western blotting has three steps, and we will very slowly go through them in order to understand. First step is that we take out the uh, entire proteins from the cell. We just slice the cell, take out every one of the proteins that we have. Next, we run what is known as a SDS page. So SDS page, as we have seen, what DNA electrophoresis is, that it separates DNA's base, like separate DNA based on the size. SDS page does something similar. It essentially separates proteins based on size. So we have separate proteins that are separated out based on their size. So heavier protein are towards the well wala side, and the lighter proteins are on the other side, the non-well side. So we have separated up the proteins, and this actually looks something like the picture we saw here. It will look like number 2, a number of bands that appear. Next, what will happen is that we will place a nitrocellulose membrane on top of the gel. So you have a gel, you put a nitrocellulose membrane on top of it, and you apply another electric field. Essentially, what happens is the proteins that were in the gel are transferred into the nitrocellulose paper. Essentially, you apply a horizontal electric field this time, the protein just transfers from the gel onto the membrane, and the membrane is positively charged, so obviously the proteins get attached to it. So now you have proteins attached to the nitrocellulose membrane, but you cannot see these proteins are not stained, so these proteins are invisible essentially. Now you do a step known as blocking, which is you add a generic protein to block the cellulose membrane. Not important. The third step, not important for our understanding, very important in the Western blotting procedure. Essentially you have transferred to the, but for us essentially you have transferred to the membrane. All the proteins from the gel has been transferred to the membrane. Now what you do is that you add an antibody against the protein that you wanted to purify. Say Prakhar wanted to purify insulin, he added only insulin, anti-insulin antibody to the gel, and the anti-insulin antibody will go and bind to insulin only, nothing else. Then we add a secondary antibody that can detect the primary antibody binding. So secondary antibody will bind to the primary antibody that has bound to insulin. So essentially you have make a chain of insulin, primary antibody, secondary antibody, and you can detect the secondary antibody like this, like bands. And this, if we see bands and how much band there, like what is the intensity of the band, we can determine what was our protein amount present in the cellular sample. This is known as Western blotting. So this is how exactly the Western blotting is done. Uh, so please tell me if you understood it. If you have any questions regarding Western blotting. This is like the gold standard in biochemistry to identify proteins. But do you have any questions regarding this? Yes, we have leaving the chat. Did everyone understand what Western blotting is? Yes, Amadip says yes. Okay. So I guess no one has any questions. So using Western blotting, we can detect our protein of interest. So that was Western blotting. So Western blotting can also be used to detect if insulin or the protein Prakhar wanted to isolate is there or not. Finally, we have something that we have. I think every one of us has an idea. Is known as ELISA. ELISA is very much used to detect presence of various substrates. You probably have heard of ELISA kits to detect malaria. Recently, there was ELISA kits which wanted to detect COVID as well, but those were not very successful. Then there are kits to detect various other pathogenic diseases such as dengue or
Hello.
AIDS.
yeah. Smriti, do you have anything to add?
Yes, sir. AIDS.
AIDS using ELISA? Yeah, you can do it. Yeah, you can. So So essentially using this particular technique, you can identify specific proteins. So we will go through exactly how it does it. So what you have is that you have a plate on which you have immobilized antibodies against a particular antigen you want to detect. What are words? Antibodies we know are essentially proteins that can capture certain other proteins, and the part they are capturing is known as antigen. So, antibody against a particular antigen, if you put the antigen into the ELISA plate, the antibody will catch hold of it. So, you have this V-shaped antibodies, blue-colored antibody on which your orange-shaped antigen has bound. Next, you add a secondary antibody which has a label on top of it, and it will also bind to the orange-colored antigen. So, essentially, you are sandwiching the antigen between your primary antibody and your labeled antibody, and now you continuously wash the entire thing. So you have the orange colored antigen attached to it. You then add our secondary antibody or the labeled antibody, and you start washing so that every one of the non specific binding of this secondary antibody gets washed off, so that only the secondary antibody that is bound to the antigen remains. And then you add what is known as a substrate that will help you detect the secondary antibody. So if after the entire process you can detect signal from the well, that means your antigen was there. And now the important thing about this is that this antigen can be anything. This antigen can be other antibodies, can be proteins, can be glycoproteins, can be hormones, anything. So using ELISA also, Prakhar can detect the presence of insulin on the protein he wanted to purify. So there are various types of ELISAs: a direct ELISA, indirect ELISA, sandwich ELISA that we just discussed, and another thing known as competitive ELISA. So this is also a gold standard in immunoassays. Immunoassays means using antibodies in order to detect various antigens. And as we just talked about, it is very much used in detection or diagnosis of various diseases. So Prakhar can use this also in order to detect his protein of interest. Is that okay? Yes, no, maybe, yeah. Is this okay that we can, what ELISA is and Prakhar can use ELISA? Yeah, okay. Ria says it is okay. Yes, okay. So that was the entire question. Archana also says yes. So that was the entire answer of the question. Prakhar can use mass spectrometry. Prakhar can use Western blotting. Prakhar can also use ELISA to detect his protein of interest. But obviously, he cannot use microarray because microarray is the full form is DNA microarray. Microarrays are used to detect the presence of DNAs, not proteins. So if you want to identify the desired protein of interest, you have to use one of the three options. Okay. So we move on and see what happened next. So post conducting the first chromatography in which got multiple bands, Prakhar said, okay, now I have to separate the bands and understand what is going on. So he wanted to purify the elution containing 4 bands that had size difference of at least 10 kilodalton. So as we saw the bands, let us go back to the bands. So as we saw, we had 1, 2, 3, and 4 bands which have a difference of more than 10 kilodalton between them. So what Prakash says is that now I have to isolate the bands, like purify the bands separately. So he must have alicot with band 1, band 2, band 3, and band 4 separate in order to identify using ELISA or mass spectrometry or Western blotting what exactly the protein is. So Prakhar thinks, how exactly can he separate the 4 bands? So he wants to use another chromatographic method, and this time he is asking what chromatographic method can be used to separate the 4 bands depending on the size difference between of 10 kilodalton. The options are reverse phase chromatography, ion exchange chromatography, affinity chromatography, and exclusion chromatography. Only one is correct out of the 4. You can go ahead and put what you think Prakar can use to separate the bands. So Archana says it is D, size exclusion chromatography. Ria also says it is D. Anyone else? Niranjana also says it is D. Okay, so we are pretty sure it is D. Okay. So, let us go ahead and look into it. And obviously, it is size exclusion chromatography. Everyone is correct. But let us know what exactly size exclusion chromatography is. Size exclusion chromatography is a chromatographic technique used to separate molecules in a solution based on their size or molecular weight. So, what happens is that you take your entire mixture that contains some large molecules and some that contain small molecules, and you put it into the size exclusion column. Now, what happens is something smart. Our column has beads in it. So the column is made out of beads, and each bead has pores inside of it. And the pores are small enough for only the smaller molecules to pass into the pores, while the larger ones cannot enter the pores at all. So after you put the sample in, what happens is that the smaller molecules trying to pass through the pores get stuck inside the beads, whereas the larger ones just flow across the column and not get into the beads at all and pass along the walls and through the space between the beads. And what that does is that the blue colored heavier or larger molecules pass through first, and then after some time you have the smaller red colored small molecules after they have traveled the entire column through the beads to pass through. So essentially, using the technique, you can separate the large molecules first, followed by the small molecules. And this is like very much used in protein chromatography as well. If you have large size difference between the proteins, you can just use this technique. The larger bands will come first and the smaller bands will come next, and you have one alicot ready at this time, you collect this, and second alicot ready at this time and you collect this, and you have separated out your bands or proteins depending on their size. Is that okay? That is fine, right? Yeah, okay. Yes. So, post conducting the first chromatography, Prakhar observed that the purified elution contains 4 prominent bands at size difference of at least 10 kilodalton. Which of the chromatographic methods can he use to obtain pure protein? The options are reverse phase chromatography, ion exchange, affinity chromatography, and size exclusion. The answer is size exclusion chromatography. That was that. We move on to the next one, which is the heavy question. So, this question is based on Sanger sequencing that was discussed during the course videos. So Prakhar needs to confirm the sequence of the recombinant plasmid he prepared. So after the entire thing, he has his protein. Uh, this is before he started even. So what he did is that he has another sequence of interest or gene of interest that he has, and he wants to confirm if that gene of interest is indeed insulin or not. So he used Sanger sequencing of DNA molecules to sequence the gene of interest, that is insulin. And the question is, which of the following is correct for Sanger sequencing? Like, which is the correct option among these? Options are 4' OH is present, and this extends an already present DNA fragment, giving a radioactive signal. 4' OH is present, terminates DNA strand from extension and gives fluorescence. 3' OH is absent, terminates DNA strand from extension, gives fluorescence. 3' OH is absent, extends the DNA fragment, gives chemical signal. Only one option is correct. Archana just goes ahead and says that it is C, that is 3' OH is absent, and this terminates the DNA strand from extension, giving fluorescence. Okay, so anyone else that wants to try and answer this? Niranjana also says C. 3' OH is absent, terminates DNA strand from extension, and gives fluorescence. Anyone else wants to try and answer this? Okay. So let us try and understand what Sanger sequencing is to start with, to understand what exactly is going on. So we have this video to play in order to understand what exactly is going on. And yeah, please don't. Yeah. Please, if you can hear the
Let's go back to the basics and explore the technology platform that has been regarded as the gold standard for many years.
You can hear the audio, right?
Yes, sir.
Okay.
Oh, hey, let's go back to the basics and explore the technology platform that has been regarded as the gold standard for many years. Yeah, you guessed it. I'm talking about Sanger sequencing by capillary electrophoresis. Many might ask, why is it called Sanger sequencing? Well, Sanger sequencing is named after the inventor of this groundbreaking technology, Dr. Frederick Sanger, who I like to call Fred, who developed this method over 40 years ago in the mid-70s. So what are the basics of Sanger sequencing? Let's take a look at our lab book. It all starts by having a short primer binding next to the region of interest. In the presence of the 4 nucleotides, the polymerase will extend the primer by adding on the complementary nucleotide from the template DNA strand. To find the exact composition of the DNA sequence, we need to bring this reaction to a defined stop that allows us to identify the base at the very end of this particular DNA fragment. Sanger did this by removing an oxygen atom from the ribonucleotide. Such a nucleotide is called a dideoxide nucleotide. This is analogous of throwing a wrench into a gear. The polymerase enzyme can no longer add normal nucleotides into this DNA chain. The extension is stopped, and we now need to identify what it is. We identify the chain terminating nucleotide by a specific fluorescent dye, 4 specific colors to be exact. Sanger sequencing results in the formation of extension products of various lengths terminated with dideoxide nucleotides at the 3' end. The extension products are then separated by capillary electrophoresis, or CE. The molecules are injected by an electrical current into a long glass capillary filled with a gel polymer. And during CE, an electrical field is applied so that the negatively charged DNA fragments move toward the positive electrode. The speed at which the DNA fragment migrates through the medium is inversely proportional to its molecular weight. This process can separate the extension products by size at a resolution of 1 base. A laser then excites the dye-labeled DNA fragments as they pass through a tiny window at the end of the capillary. And the excited dye emits a light at a characteristic wavelength that is detected by a light sensor. Software can then interpret the detected signal and translate it into a base call. When the sequencing reaction is performed, in the presence of all 4 terminated nucleotides, you eventually get a pool of DNA fragments that are measured and separated base by base. What you will get in the end is a data file showing the sequence of the DNA in a colorful electropherogram, and a text file which you can use to answer the questions you may be asking. And that, in a nutshell, is Sanger sequencing. For more
Yes, so that is the video. So
information,
we will go once through this and then we can have a discussion. So essentially our normal DNTP or deoxynucleotide looks something like this, with a ribose ring and a nitrogenous base. It can be ATGC attached here. Our ribose ring has a 3' OH, but does not have a 2'. It has a 3' OH, and that is why it is known as deoxynucleotide. It does not have a 2' OH. That is why it is known as deoxynucleotide. So it has only a 3' OH, and this 3' OH is very important to add a next nucleotide to the entire chain. So this nucleotide will get attached here. So next nucleotide will attach, like A will attach to the next nucleotide, which can be a G, via this 3' OH. So if we want to stop the sequencing, then we add something known as a dideoxynucleotide, which does not have a 3' OH. So when this gets incorporated into the DNA strand, the DNA sequencing just stops. DNA polymerase just stops and cannot add anything more to the strand. So this process is used in the entire technique of Sanger's DNA sequencing to sequence the DNA template. So the things that we need for Sanger sequencing is the template DNA. We need the dideoxy nucleotides, the DD, ATP, TTP, GTP, and CTP. Our normal DNTPs, a polymerase, and a primer. What happens is that our primer binds, polymerase starts extending the replicated chain or the complementary strand to the template, and while doing this, a DDNTP can be incorporated into the elongated chain. Once DDNTP gets into the chain, the reaction just stops and no more extension can occur. And this DDNTP is something that we have to know is fluorescent in nature. So what we have is that at the end of the entire reaction process, we have at the end of the entire reaction process is that multiple strands or multiple lengths of the complementary template which has a DDNTP attached at the end. So we have a color coded we have color coded lengths of DNAs which are complementary to our starting strand. Now we only have to run this entire mixture through a capillary gel electrophoresis, which is similar to gel electrophoresis, just that it can separate the DNA sample on a one nucleotide level, and depending which sample passes first. So obviously the smallest one will start pass first, and thus we will have the chain with the only one nucleotide added to its end. That is here in this case T, which is a fluorescent T, which will give us a red signal. So this will pass first, followed by a carrying strand, and then we know that the next one is A, next a G, next a C, next a T again, next a G, and thus the sequence of flow will determine the sequence of the complementary DNA strand. And this is the entire thing is calculated in this entire thing is calculated and computed in a CCD camera, and then we have an output which is known as an electropherogram, which is essentially a graph type description of the entire sequencing reaction, and we will have the separate peaks that will give us what exactly the sequence was. So this is entire thing is known as Sanger DNA sequence. So if anyone has any questions regarding this. So you can go ahead and ask what if you have any questions regarding Sanger DNA sequencing. So everyone understood everything that we had in this, right? Yes, no. Okay, Amardeep says yes. Ria also. Ria says no. Okay. So we have a no. So I will go through the entire thing. So what we have is that for Sanger sequence. So in order to understand what the sequence of the template is, you can use you have to stop the replication process of a complementary strand at one stop, and when it stops you identify what nucleotide was added. So say a replication is going on and a was added and you stop the reaction just then, you take a snapshot at that stage, so you know that a is added, so obviously the complementary one should be t. So our template has a t inside. But how exactly do you stop the reaction when you want it? Sanger did it using a DDATP or a dideoxynucleotide. When a dideoxynucleotide is incorporated in a primer chain, the entire reaction is stopped, essentially giving you a snapshot of the replication. So what happens is that you do a DNA replication process, or the DNA replication essentially. You have a polymerase, you have a primer, and you allow the primer to start the DNA replicase enzyme to start at DNA polymerase. It should be polymerase, DNA polymerase to start adding DNTPs at the one end. Once the DNTPs are being added, at one point there will be a DDNTP that is getting incorporated into the chain. Once that is incorporated, the entire thing stops. Once we have a stop, that means that once this A is incorporated, thus the complementary template sequence should be a T. But thus if we can get hold of all the stops, then we can actually calculate, like detect, compute what the complementary strand sequence was. So when you do the entire reaction with the DDNTPs involved in it and take the mixture out, you have a mixture of DNA which looks something like this. So you have your primer at to which at one end only one DDNTP, that was a DDT, was added. The chain annealing will start from the 3' OH. The chain extension will start from the 3' OH. Yes, you are correct. But since our DDNTPs do not have a 3' OH, so they will not allow a next nucleotide to be added, but they will be incorporated. Like they will get and bind into the they will be inserted into the new strand, but will not allow anything else to be bound after. So this final DDNTP is the final stop. No more chain extension for this particular chain. So if we had a so when you look at the entire mixture, what you will see is that the smallest chain will contain only one nucleotide that was added to its end, and that was unfortunately a DDNTP. And that is why after only one base addition, the entire reaction stopped. The next one will be a second DDNTP. Like first one was a normal DNTP, second was a DDNTP that stopped the reaction. So you have a T at the end, and the next will be a A. Similarly, you have the entire mixture. So one after the other at individual bases, the DDNTPs will be incorporated, and you will have a mixture of DNAs which are fluorescently labeled, and will have a DDNTP at its end, one after the other. When you run the entire thing in capillary electrophoresis, you will see the smallest band come the first, and you will detect the signal, and you will say, I see a T here. That means the first should have been a T that was incorporated. Next, you will see a and then you will say the next one that was incorporated was a, and that is why the signal goes T A, then G, then C, then T, then G, and this is how you calculate the entire sequence of the complementary strand. And this is what is detected and displayed to you on the monitor. Was that okay? No. Yes is okay. Okay, I guess. Like we can try. I can try drawing this and explaining if you want. There you go. Is this okay with everyone, or we can go through this again? Yes, no, you can go ahead and answer this. Like put in the chat box if this is okay with everyone or not. Okay. So if it is clear and no one has any questions, that we do not probably need to delve into this. Okay then. So this is what Sanger sequencing is, using dideoxynucleotides which are fluorescently labeled in order to detect the sequence of a DNA. So looking at the diagram, obviously we know that the dideoxynucleotide that is used here does not have a 3' OH, and that is why the third option is correct. That is, using the Sanger sequencing method, the 3' OH is absent and terminates DNA strand from extension, giving fluorescence. So we go on to the next question. Prakar isolated recombinant plasmid for sequencing and diluted sample with a dilution factor of 1:40. The UV visible spectrophotometer absorbance reading at 260 and 280 nanometer, obtaining 0.42 and 0.21 readings respectively. Calculate the concentration of the dilute and the concentrated plasmid samples. So the options are given: plasmid DNA concentration in plasmid 21 nanogram per milliliter, DNA concentration is plasmid diluent 67.2 nanogram per milliliter, and they just continue. So yeah, so essentially this is a mathematics problem. We can probably, so I would encourage everyone to try and like calculate this. I will even give you the formula for this. So since probably we need a refresher on what the formulas for DNA absorbance is. So if DNA absorbance is easily calculated by estimating the absorbance at 260 nanometer. So A260 is the important factor here. So we do not care about the A280 regarding the concentration of the DNA. We could calculate it in order to detect the purity of the DNA. For using A260 value only, we can detect the amount of DNA present. The formula here is the A260 of one, like the absolute A260 of is 50 microgram per milliliter. So essentially your concentration of your DNA will be A260 reading into 50 microgram per milliliter. Here the A320 reading is used as a blank. We do not have that here, so we will ignore it. And dilution factor for the first one is, for this sample is 1, and then we will multiply by divide by 40 in order to calculate the dilution. So the concentration is essentially the A260 value multiplied by 50 microgram per milliliter. You can, I guess, go ahead and just calculate, just multiply by 50 and tell me what the answer should be. The 8 to 60 reading is 0.42. Using the formula, concentration is 8 to 60 into dilution factor into 50 microgram per milliliter. What should be the correct answer? Multiply by 50. Yeah, we can put into the chat which you think is the correct answer, right? You understood what I was trying to say, right? Or do you need help regarding this? So essentially what you do is like we take the A260 value, which is 0.42, and multiply it by 50 microgram per milliliter. So you get 21 nanogram per 21 nanogram per milliliter as your answer, microgram per milliliter as your answer, which is number D. and then you dilute it 40 times, that is divide this by 40, and then you get 0.52 microgram per milliliter. So your answer should be D. Mental math, this is just simple mental mathematics. These type of questions also get asked in various exams. In those cases, sometimes you will get it somewhat changed. That is, instead of a pure SSD or dsDNA, you will get a RNA. For that, the absolute value is 40 microgram per milliliter, and then vice versa. You can just go and check what the absolute values are, multiply them with the 8 to 60 reading, and get your answer. That was that. We go on to the next question. A research group decided to use a different technique in order to achieve intact insulin as a dietary supplement. So what Prakhar was doing, a separate group started doing something else. They succeeded in obtaining the desired protein in goat's milk. So which of the following is true for the above mentioned conditions? The options are: the goat is a transgenic and the gene of insulin is added into the genome of the goat. The goat ate insulin which got completely digested and came to the milk. The goat ate a vector containing insulin, which after transcription and translation formed the insulin and came in the milk. They are lying as a protein not present in the goat's genome cannot be present in the milk of a goat. Okay. So there only one option is correct out of the four. Niranjana says it is one. Goat is transgenic and gene insulin is added into the genome of the goat. Archana also says A. Anyone else that wants to try and answer this? Yeah, also says A. Okay. Okay. So you are all correct, and the other options are not very attractive, but obviously you are all correct. Amardeep also says that is the goat is a transgenic animal. So let us go and understand what a transgenic animal is. A transgenic animal is an animal that has a stably incorporated engineered DNA into a germ. So stably incorporated is a key word here, because a foreign DNA can be incorporated into a genome and then get kicked out of the genome, and that is not a transgenic animal. A transgenic animal should have a stably incorporated engineered DNA in its genome. A foreign DNA that was not present in it, you put it into the genome from outside, and it gets stably incorporated in it such that it is inheritable in nature. That is, the goat will pass on the gene, the foreign gene or the recombinant gene in its progeny also. Such an organism is able to pass the transgene onto all offspring. So this is known as a transgene. The foreign gene that you have incorporated is known as a transgene. It should be stressed that all cells of a transgenic individual contains the transgene. So in a transgenic animals, all the cells in the animal should have a transgene. Should have the transgene that you have incorporated. So how exactly is this done? So there are many methods. We will discuss those. One of those include taking the human gene, taking an egg from the cow or whatever the animal is. It is goat. Okay, taking a egg from the goat and incorporating the gene into the egg via microinjection and allowing this egg to develop into a fully grown goat. And after the development of the goat host, this goat has the transgenic gene in it, and the milk from it should be transgenic in nature, and which, if engineered correctly, will carry insulin. So transgenic animal has the transgene incorporated in all its cells, this causes it to express the transgene and form the protein that you wanted. How exactly is it done? It is done via three methods. The first is known as a pro-nuclear injection, in which you take your DNA construct, put it into a fertilized egg, and implant it into a female recipient, which is the surrogate mother. This will give rise to a particular, after giving rise to the offspring, will have a transgenic offspring present in the whole litter. Another method is using embryonic stem cells. You take your DNA construct, you allow for homologous recombination or just addition of the DNA construct into something known as a blastocyst. So blastocyst is a cell that is extracted from the fertilized egg at an early stage, and this blastocyst is then implanted into a female recipient, and the process is the same. This blastocyst then forms the entire fertilized egg, which will give rise to number of progenies. All these progenies will have part of their genome, the part of their entire body of cells as the transformed cell. So in this process, the entire, in the first generation only, you do not have all the cells that are getting transferred into the transgenic line. You will have some cells which are shown here as black, which are transgenic. The white are the normal mice cells. What you do is that then you take out of all of these one mice which has the transgene present in its germ line or germ cells. This is in this case we say that the particular mutation, a particular transgene has gone germline. That means that when these mice will give rise to another progeny, then the germ cells will get transferred, will transfer the only the transgene to the next progeny. So essentially what you do is that take a DNA construct, add it to the blastocyst cell, add the blastocyst cell to the female recipient, form chimeras. Chimeras have both type of cells. Find a chimera that has a modification in the germ cell, have the germ that chimera give rise to new litters, and after selection you will have the entire mice having the just the transgene. And something, the last is known as nuclear transfer. So in nuclear transfer, you have the DNA construct, add it to cultured somatic cells. These are not germline cells. Somatic cells are normal cells that are present in our body. Take a fertilized egg, take the nucleus out, put the somatic cell nucleus in, implant this into a female recipient, allow this to form and like grow into a new animal that will have the transgene. So this is known as somatic nuclear transfer. This is also a cloning procedure. Using this technique only, we had our first cloned animal, which was Dolly, the cloned sheep, and she was created using somatic nuclear transfer. Essentially, the entire slide just tells you that we can have foreign DNA or foreign genes being expressed in a eukaryotic animal, and that animal is known as a transgenic animal, and the particular transgene is incorporated into every cells of the animal, and now that animal will produce or express the protein of your interest. So the correct option is one. The goat is transgenic, and the gene insulin, if you want to express it into the goat milk, is incorporated into the goat genome, and thus the option one is correct. Does anyone have any questions regarding this particular question? Probably not. So this was probably a bit difficult slide to understand. Maybe you can go ahead and go through the lecture material on transgenic animals, and and go through the book as well, the recommended book. And if you have any questions, you can just go ahead and put it into the discussion forum of NPTEL, and we can discuss it. This is a difficult part to understand. But you have any questions, we can answer there. So the similar things that we studied in the course was transgenic plants, how they are created, but this is just for transgenic animals. So this is the last question of the day. Given the table are few components of mRNA isolation in column A and roles of the biomolecules in column B. You have to choose essentially they match the following between column A and column B. So this was shown in the laboratory demonstration session, which part like which component is responsible or used for which function. So column A, first we have RNA ZAP, we have EDTA, we have DNAs, we have trizol, and in column B we have the functions of these particular things. First is degrade genomic DNA contamination, degrade mRNA contamination, prevent DNA degradation, prevent RNA degradation, prevent RNA hydrolysis, increase RNA hydrolysis, maintain cell integrity, lyse cells, and bring out contents. And we have separate various choices matching various ones. So like look at the particular columns and try and solve this. Only one is correct, obviously, because only one option type of correct answer this is. We can go ahead and try and answer this one. Amardeep says C. Archana also says C. Anyone else wants to try and answer this? Okay, okay. So let us just quickly go through what does what. So RNA is so during RNA extraction, as we just discussed throughout the entire session, RNA is very labile and easily degraded. So you should, in order to protect RNA or during mRNA isolation, you have to make sure there is no RNase enzyme present anywhere around, and that is done using something known as RNase zap. So RNase zap, the function of it is to completely remove RNase contamination from glass and plastic surfaces, and this essentially prevents RNA degradation by removing RNase enzyme. EDTA is a chelating agent which chelates out magnesium. So this is also protection mechanism for RNA. So there are enzymes that are known as hydrolases that also degrade RNA, but they need the magnesium 2 plus ion in order to degrade RNA. So EDTA acts as a chelating agent which chelates out all the magnesium 2 plus ions present in a solution. Essentially, it takes all the magnesium ion and keeps it to itself. So no magnesium ions are there in the solution for hydrolysis enzymes to work, and thus this prevents RNA hydrolysis. Then we have DNAs. So in the entire procedure of mRNA purification, there will be a DNase component attached to it. So DNA will try and get into the final mixture. You do not want that. You want to remove all the DNA that is present. So you add the DNase, which degrades all the DNA present in the mixture. So DNA gets off, gets removed, and you only remain with your pure mRNA. So this removes genomic DNA contamination. And then you have something known as trizol. So trizol is, I guess, very important in these kind of preps. So what trizol is a ready to use reagent for isolation of high quality total RNA or RNA DNA protein from a variety of sites. Well, what is trizol? Trizol is essentially nothing very fancy. It is a solution in which you put your cells into, and what it does is first it lyses the cell. It completely disrupts the cell and brings everything out. And when you centrifuge trizol, what will it do is that it will form three layers, three separate layers it will form. And the three layers, one layer will have the protein of the cell, one layer will only have the RNA of the cell, one layer will have only the DNA of the cell. And this is very, very useful in order to do preps for RNA isolation, because this reagent not only lyses the cell but also separates all the components for you to easily isolate. So, so the essential work of triazole is to lyse the cells, bring out the components, and also separate the all the components based on what type of component it is. So, using this particular chart, we can easily solve the question. RNA zap is used for preventing RNA degradation. EDTA used for preventing RNA hydrolysis. DNA is used for degrading genomic DNA contamination. Trizol used for lysing cells and bringing out components. So you were correct. It is number C. So that was the last question that we had to deal with today. Thank you for everyone for joining. If no one has any other questions, then we can stop here. So this is the YouTube link on which every one of the recorded sessions are being uploaded. So if anyone wants to go through these sessions again, or you have not attended maybe the sessions and want to watch the sessions, then could not attend the sessions live, you want to watch it again, you can do it using the YouTube link. Okay. So if no one has any other questions, I thank you all for joining today's session. See you all next week. Thank you.